I am a clawed lobster of the family Nephropidae, and more specifically, I belong to the genus Homarus. My kind has walked the ocean floor for more than 100 million years, long before the continents took their present shapes, long before the first human ever glimpsed the sea. Scientists place me within the phylum Arthropoda, the most diverse assembly of life on Earth, and within that vast collection I belong to the subphylum Crustacea, sharing ancestry with crabs, shrimp, crayfish, and barnacles. Within Crustacea, I am a member of the class Malacostrica and the order Decapita, meaning 10 legs, though that count includes my claws, which are legs transformed into tools of survival. My family, Nephropidae, contains the true clawed lobsters, distinguished from spiny lobsters by the presence of those massive chelae, the claws that define my silhouette and my way of life. There are 2 species in my genus that most people know. I may be Homarus americanus, the American lobster, ranging from Labrador to North Carolina along the western Atlantic, or I may be Homarus gammarus, the European lobster, found from the Norwegian coast down to the Mediterranean and along the Atlantic shores of Europe and North Africa. The 2 of us are similar in form and habit, separated by an ocean and by subtle differences in color, size, and the fine details of our genetics. For the purposes of this account, I will speak as Homarus americanus, but much of what I describe applies to my European cousin as well, and I will note the differences when they matter. My body is a masterpiece of segmented armor. From the tip of my rostrum, the pointed projection between my eyes, to the end of my tail fan, I may measure anywhere from 20 centimeters to over 1 meter in total length, though most of my kind fall between 25 and 60 centimeters. My weight varies enormously with age and food availability. A young adult might weigh half a kilogram, while the largest individuals on record have exceeded 20 kilograms, though such giants are vanishingly rare now. My body is divided into 2 main sections: the cephalothorax, a fused head and thorax covered by a single hard shell called the carapace, and the abdomen, which most people call the tail. The carapace is smooth and slightly arched, mottled in shades that blend dark greenish blue, brown, and sometimes hints of orange or red. This coloration comes from a complex of pigments bound to proteins in my shell, and it serves to camouflage me against the rocky, weedy, and muddy substrates where I spend my life. The famous bright red of a cooked lobster is not my living color. That transformation happens when heat breaks down the protein complexes, releasing the red pigment astaxanthin that was always present but masked by other hues. My carapace is not a single static shield. It is composed of chitin, a polysaccharide that forms a tough, flexible matrix, hardened by calcium carbonate and other minerals. Beneath the carapace, my body is segmented, a heritage of my arthropod lineage. My abdomen consists of 6 distinct segments, each overlapping the next, allowing me to flex and curl my tail with precision. At the end of my abdomen is the telson, a central plate flanked by 4 uropods, flat appendages that together form my tail fan. This fan is my emergency escape system. When threatened, I contract the powerful muscles of my abdomen in a rapid flexing motion, snapping my tail beneath my body and propelling myself backward through the water in a series of quick thrusts. This escape response, called lobstering or the charadoid escape reaction, can carry me several body lengths in seconds, a blur of motion that often startles predators long enough for me to reach a crevice or burrow. My head bears a pair of long antennae, often as long as my body or longer, and a pair of shorter antennules, which are branched and house chemoreceptors. The antennae are sensory organs and also serve as tactile probes, sweeping the water and the ground ahead of me as I walk. My eyes are compound, mounted on short stalks that can move independently. Each eye is made up of thousands of tiny units called ommatidia, giving me a mosaic view of the world. My vision is not sharp by vertebrate standards, but I am sensitive to movement and changes in light, and I can detect the polarization of light, which helps me navigate and orient myself in the water. I see best in the blue-green spectrum, the wavelengths that penetrate deepest in the ocean, and my vision is adapted to the dim, shifting light of the seafloor. Beneath my carapace, my body is a crowded cavity of organs. My heart is a compact muscular chamber located dorsally, pumping hemolymph, my blood, through an open circulatory system. My hemolymph is pale and translucent, tinted faintly blue when oxygenated, because it carries copper-based hemocyanin rather than iron-based hemoglobin. My gills are feathery structures located in chambers on either side of my thorax, beneath the carapace. Water flows over them continuously, drawn in at the base of my legs and expelled near my mouth by the beating of specialized appendages called scaphognathites. These gill balers create a current that brings oxygen and carries away carbon dioxide and metabolic waste. I extract oxygen efficiently even in cold water, where it is more abundant, and my metabolic rate is tuned to the temperature of my environment. My digestive system begins at my mouth, a complex arrangement of mouth parts that manipulate and shred food. I have mandibles for crushing, maxillae and maxillipeds for handling and sorting, and all of these work in concert to process my prey before it enters my esophagus. The esophagus leads upward, yes upward, into my stomach, which is located in my head just behind my eyes. My stomach is divided into two chambers, the cardiac stomach, where food is ground by a set of hard chitinous teeth called the gastric mill, and the pyloric stomach, where digestion and absorption begin. The gastric mill is a remarkable structure, 3 teeth arranged in a triangle, moved by muscles, grinding food into a paste. From the pyloric stomach, a pair of digestive glands, often called the hepatopancreas or tamale, secrete enzymes and absorb nutrients. Waste passes through a long intestine that runs the length of my abdomen and exits through the anus, located ventrally on the telson. My nervous system is built on the arthropod plan, a ventral nerve cord running the length of my body, with ganglia, clusters of nerve cells, in each segment. My brain is a collection of fused ganglia in my head, processing sensory input from my eyes, antennae, antennules, and the many sensory hairs, called setae, that cover my body. These setae are sensitive to touch, water movement, and chemical signals. I am covered in chemoreceptors, particularly dense on my antennules, mouth parts, and the tips of my walking legs. I taste the water constantly, detecting the faintest traces of amino acids, sugars, and other molecules that signal food, predators, or mates. My sense of smell, or more accurately my chemical sense, is acute and directional. I can follow a scent plume upstream to its source, triangulating the location of a dead fish or a potential mate from meters away. My 10 legs, the decapod signature, are not all alike. The first pair, my claws, are greatly enlarged and specialized. In most individuals, the 2 claws are asymmetric. One, called the crusher claw, is heavy and blunt, with rounded tubercles on the closing surfaces, designed to crack shells and crush hard prey. The other, the cutter or ripper claw, is more slender, with sharp-edged teeth, used to tear flesh and manipulate softer food. This asymmetry develops during my youth and is not strictly linked to left or right. Some of us are left-clawed, some right-clawed. The crusher claw is powered by enormous muscles, and the force I can generate is formidable, measured in hundreds of newtons, enough to break through the shells of clams, mussels, sea urchins, and crabs. The remaining 4 pairs of legs are walking legs, or periopods, each ending in a small pincer or simple point. I use these to walk, to probe the substrate, to groom my antennae and body, and to manipulate objects. My walking gait is slow and deliberate, a forward and sideways shuffle, and I can move in any direction, though forward is most common. I live on the bottom. My habitat is the continental shelf, from the intertidal zone, where the tide exposes rocks and pools twice a day, down to depths of approximately 480 meters, though I am most abundant in water less than 50 meters deep. I prefer substrates that offer shelter, rocky outcrops with crevices, boulder fields, cobble, and areas where I can dig a burrow beneath a stone or wedge myself into a crack. I avoid wide expanses of soft mud or sand where there is no cover, though I will cross such areas at night if necessary. My European relative, Homarus gammarus, shows similar preferences but is often found in somewhat rockier and more complex habitats on average, and tends to occupy deeper water less frequently than I do. The water temperature shapes my world. I am ectothermic, meaning my body temperature matches the surrounding water, and my metabolic rate, growth rate, and activity level are all temperature dependent. In the northern parts of my range, off Canada and Maine, summer water temperatures may reach 12 to 18 degrees Celsius, while winter temperatures can drop to near zero or slightly below. In the southern parts of my range, off southern New England and the mid-Atlantic states, summer temperatures can exceed 20 degrees Celsius, and winters are milder. I am most active when water temperatures are between 10 and 20 degrees Celsius. Below 5 degrees, I become sluggish, my feeding declines, and I may remain in my shelter for extended periods. Above 20 degrees, I face stress. My oxygen demand increases, but the water holds less dissolved oxygen, and I become more vulnerable to disease. Salinity matters too. I live in full-strength seawater, typically 30 to 35 parts per thousand, and I am sensitive to freshwater intrusion. My gills and body surfaces regulate ion balance, but I cannot tolerate prolonged exposure to low salinity. Estuaries and river mouths are generally poor habitat for me unless the mixing is weak and salinity remains high. I am nocturnal by preference, though this is not absolute. During the day, I usually remain in my shelter, a burrow I have excavated beneath a rock or a crevice I have claimed. I face outward, antennae extended, claws ready, monitoring the water for threats and opportunities. My shelter is critical. It protects me from predators, provides a stable microenvironment, and serves as a base from which I forage. I may occupy the same shelter for weeks or months, or I may move frequently, depending on food availability, competition, and disturbance. If another lobster challenges me for my shelter, we may fight, displaying, pushing, and grappling with claws. The outcome usually depends on size. The larger individual typically wins and claims or retains the shelter. Injuries from these fights can be severe, with claws broken or lost, though I can regenerate a lost limb over several molts. As evening approaches and light fades, I emerge. I leave my shelter cautiously, antennae sweeping, eyes scanning for movement. I walk slowly across the bottom, following scent trails, investigating crevices, probing under rocks and among seaweed holdfasts. I am hunting. My diet is omnivorous and opportunistic. I prefer live prey when I can catch it. Crabs, small fish, marine worms, sea urchins, sea stars, mollusks such as clams, mussels, snails, and occasionally other lobsters. I also consume carrion, dead fish, dead invertebrates, anything with protein and calories. I am not above scavenging, and my sense of smell leads me efficiently to carcasses. When I find a mussel or clam, I use my crusher claw to crack the shell, sometimes wedging it against a rock for leverage. Then use my cutter claw and mouth parts to extract and shred the soft tissue. When I catch a crab, I may crush it outright or tear off limbs and consume them one by one. I also eat algae and other plant material, scraping it from rocks, and I ingest organic detritus, the fine particulate matter that settles on the bottom. This varied diet makes me an important generalist in the coastal ecosystem, neither strictly predator nor scavenger but something in between. My feeding activity peaks in the hours after dusk and before dawn, though I may feed at any time if a strong scent reaches me. In summer, when water is warm and my metabolism is high, I feed nearly every night. In winter, when I am sluggish, I may feed only once or twice a week. My feeding rate is also influenced by molt cycle. Just before and during molting, I do not feed at all, and immediately after molting, my new shell is too soft to crack hard prey, so I focus on softer food or wait until my shell hardens. My growth is not continuous but occurs in discrete jumps. To grow, I must molt, shedding my entire exoskeleton and emerging in a new, larger shell that is initially soft and flexible. This process is called ecdysis. In preparation for a molt, I reabsorb calcium from my old shell and store it in my digestive glands and in structures called gastroliths, which are small calcium-rich deposits in my stomach. Hormonal signals, controlled by glands in my eye stalks and thorax, trigger the molt. I stop feeding days or weeks in advance and I retreat to my shelter. The old shell splits along a seam on my back and I pull myself out, legs first, then body, then claws, and finally my antennae and eye stalks. The entire process takes minutes to hours, and it is exhausting and dangerous. My new shell is pale, soft, and wrinkled. I take up water to expand my body and stretch the new shell to its full size. Over the following days and weeks, my shell hardens as I deposit chitin and remineralize the cuticle with calcium carbonate. During this period, I am extremely vulnerable. I cannot defend myself, I cannot crush prey, and I am easy prey for fish, octopuses, and other lobsters. I remain hidden, sometimes for a week or more until my shell is hard enough to function. Young lobsters molt frequently, perhaps 25 times in the first 5 to 7 years of life. As I age, my molting frequency declines. A mature adult may molt only once a year, or once every 2 years, or even less frequently. Each molt allows growth, but also carries risk. A small percentage of lobsters die during the molt, trapped in the old shell, or are killed by predators immediately afterward, or succumb to infection or injury. The energy cost of molting is enormous, and if food has been scarce, I may not have the reserves to complete it successfully. Molting also allows me to regenerate lost limbs. If I lose a claw or a leg in a fight or accident, a small bud appears at the site during the next molt, and with each subsequent molt, the limb regrows, usually reaching full size after 3 or 4 molts. This regenerative ability is vital, as limb loss is common in my life. However, there is a cost. Regenerating a large claw diverts energy from body growth and reproduction, and a lobster with a regenerating limb may grow more slowly overall. My lifespan is long. In the wild, I may live 50 years or more, possibly much longer. There are reports of lobsters estimated to be over 100 years old, though aging us is difficult. The most reliable method involves counting growth bands in the eye stalk or gastric mill, similar to counting tree rings, but this requires sacrificing the animal, and the technique is still being refined. Mortality is high in early life, but if I survive to adulthood and avoid predators, disease, and human harvest, I can live for decades. There is some debate among scientists about whether lobsters experience senescence, the biological aging seen in most animals. Our mortality rate does not appear to increase strongly with age, and we continue to reproduce throughout life, though some evidence suggests very old lobsters may experience reduced reproductive success and increased vulnerability to shell disease. The question is still being studied. I do not live in permanent social groups. I am largely solitary, territorial, and often aggressive toward other lobsters. However, I am not entirely antisocial. My interactions with others of my kind are frequent and complex, governed by a mix of chemical signals, visual displays, and direct physical combat. When two lobsters meet, there is usually a confrontation. We approach each other, antennae extended, and jets of urine from openings near our antennules are released. This urine contains chemical signals that convey information about size, sex, reproductive status, and individual identity. In many cases, one lobster, usually the smaller, will retreat without a fight, recognizing the dominance of the other. If neither retreats, the confrontation escalates. We may engage in a ritualized display, raising our bodies, extending our claws, and spreading our antennae and tail fan to appear larger. We may lunge, grapple, and push. If the display does not resolve the contest, we fight in earnest, claws locking, each trying to flip or injure the other. These fights can result in lost limbs, eye damage, or punctured carapaces. The winner gains access to the contested resource, a shelter, a feeding area, or, in the case of reproductive encounters, a mate. The loser retreats and usually avoids the winner for days or weeks, recognizing the individual by chemical signature. Dominance hierarchies can form in areas of high lobster density, where the same individuals encounter each other repeatedly. A large, successful male may dominate a local area, claiming the best shelter and excluding rivals. Subordinate lobsters learn to avoid him, moving through his territory cautiously or at times when he is inactive. These hierarchies are not rigid, and they shift as lobsters molt, grow, and change size relative to one another. A subordinate that grows and molts successfully may challenge and defeat a former dominant, reversing their roles. Females, too, establish and defend territories, particularly when they are carrying eggs. A female with a good shelter and access to food will defend her space vigorously against both males and other females. However, reproductive dynamics introduce a different pattern. When a female is ready to mate, her chemical signals change. She seeks out a dominant male, approaching his shelter and signaling her receptivity with urine-borne pheromones. The male, who moments before might have attacked an intruding lobster, becomes tolerant, even protective. The female enters his shelter, and they cohabit for days or weeks. This cohabitation is unusual in our species and appears to be a strategy that allows the female to mate safely while she is soft and vulnerable after molting. Mating is tied to the female's molt cycle. A female can only mate when her shell is soft, immediately after molting, because the male must insert his spermatophores, packets of sperm, into her body through openings on her underside, and this is only accessible when she is soft. The mating sequence begins with courtship. The receptive female approaches the male's shelter repeatedly over several nights, each time releasing pheromones and engaging in ritual displays, sparring gently with claws, touching antennae, and sometimes engaging in a behavior that looks like boxing, where they push at each other with open claws. The male may initially respond aggressively, but the female's chemical signals and persistence eventually trigger a shift in his behavior. He allows her into his shelter. She molts inside the shelter while he stands guard. Once she has shed her old shell and is soft, they mate. The male turns her onto her back and deposits spermatophores onto the underside of her thorax, near the openings of her oviducts. The sperm are stored in a seminal receptacle inside her body, where they remain viable for months. After mating, the female stays in the male's shelter for a period of days, often about 2 weeks, during which her shell hardens. The male protects her during this time, fending off other lobsters and predators. Once her shell is hard, she leaves and returns to her own territory or establishes a new one. The male may then mate with another female if one approaches, and this pattern can repeat through the breeding season. Females, in contrast, mate only once every 2 years or less frequently, tied to their reproductive cycle. After mating, the female does not lay eggs immediately. Sperm remains stored in her seminal receptacle for months, sometimes up to a year. When she is ready, she extrudes eggs from her ovaries, which are located in her cephalothorax. The eggs pass through her oviducts, are fertilized by the stored sperm as they exit, and are attached to the underside of her abdomen. She carries the eggs externally, glued to specialized appendages called pleopods, which are small swimming legs under her tail. A female may carry anywhere from 5,000 to over 100,000 eggs, depending on her size. Larger, older females produce more eggs. The eggs are dark green when first laid, each about 1.5 millimeters in diameter, and over the months of incubation they darken, eventually turning nearly black as the embryos develop and their eyes become visible through the egg membrane. This is why egg-bearing females are often called berried lobsters. The egg mass resembles a cluster of dark berries. The female cares for her eggs with remarkable diligence. She fans them regularly with her pleopods and tail, creating a current of water that delivers oxygen and carries away waste. She grooms them with her small rear legs, removing debris and picking off any eggs that die or become infected with fungi or bacteria. This grooming is critical. Without it, fungal infections can spread through the egg mass and kill the entire brood. The incubation period is temperature dependent. In cold water, incubation may last 9 to 12 months. In warmer water, it may be as short as 6 months. Throughout this time, the female continues to forage, though her movements may be more restricted and cautious. She is more vulnerable while carrying eggs, both because she is more conspicuous and because she cannot perform the rapid tail flip escape as effectively with a mass of eggs beneath her. When the eggs are ready to hatch, the female seeks a suitable location, often an area with good current and away from heavy predation. Hatching occurs over a period of days to weeks. The larvae emerge from the egg membrane and immediately swim upward, propelled by their pleopods and their small, flattened bodies. They are now planktonic, drifting in the open water, and they bear little resemblance to me. Each larva is about 8 millimeters long, translucent, with a long, segmented body, large eyes, and prominent spines. They are called zoea larvae, though in lobsters they are sometimes specifically referred to as lobster larvae or stage 1 larvae. The larval stage is perhaps the most perilous time of my life cycle. Larvae drift in the surface waters in the plankton for weeks to months, depending on temperature. They pass through 3 planktonic stages, each separated by a molt. In the first 3 stages, they swim actively in the water column, feeding on smaller plankton, including copepods, larval crustaceans, and phytoplankton. They are voracious feeders, growing rapidly, but they are also food for countless predators: fish, jellyfish, ctenophores, seabirds, and other planktonic organisms. Mortality in the larval stages is extremely high. It is estimated that fewer than 1 in 1,000 larvae survive to settle on the bottom. Currents carry larvae away from their hatching site, sometimes for hundreds of kilometers. This dispersal is important for genetic mixing and colonization of new habitats, but it also means that most larvae are lost to unsuitable areas or predators. After the 3rd planktonic molt, a larva enters the 4th stage, called the post-larva or stage 4 larva. This stage marks a transition. The post-larva still swims in the water column, but it looks more like a tiny adult lobster with claws and a tail, though it is still only about 12 millimeters long. The post-larva begins to exhibit a behavior called exploratory diving. It swims down toward the bottom, explores the substrate, and then swims back up. It repeats this cycle, sometimes many times, until it finds a suitable place to settle. Suitable substrate is critical. The post-larva seeks out cobble, small rocks, or complex structure with numerous tiny crevices where it can hide. If it settles on open sand or mud, it is almost certain to be eaten. Once it finds good habitat, it settles and undergoes another molt, becoming a juvenile lobster approximately 15 millimeters long. It is now benthic, living on the bottom, and will remain so for the rest of its life. The early juvenile phase is a time of hiding. I am tiny, vulnerable, and surrounded by predators. I remain in crevices and under small rocks, emerging only briefly to feed on detritus, tiny mollusks, and other small invertebrates. I molt frequently during this period, every few weeks, growing with each molt. As I grow, I require larger shelters and larger prey. By the time I am a few years old and several centimeters long, I resemble a miniature version of my adult self, and I begin to behave more like an adult, foraging more widely, defending small territories, and interacting aggressively with other juveniles. Sexual maturity is reached at a size that varies with water temperature and population. In colder waters, I may not mature until I am 6 to 8 years old and weigh around 400 to 500 grams. In warmer waters, maturity may come earlier, at 4 to 6 years. Males typically mature slightly earlier and at a smaller size than females. Once mature, I participate in the reproductive cycles I have already described, and I may continue to reproduce for the rest of my life, which could be decades. My role in the ecosystem is multifaceted. As a predator and scavenger, I influence the abundance and behavior of many other species. I consume large numbers of sea urchins, and in some areas, my predation helps control urchin populations, which in turn affects the abundance of kelp and other seaweeds that urchins graze. I eat crabs, mollusks, and worms, affecting their population dynamics. I compete with other bottom-dwelling predators such as cod, sculpin, and other fish for food and space. I am also prey. Juvenile lobsters are eaten by a wide range of fish, including cod, haddock, flounder, wolffish, and sculpins. Octopuses are significant predators of lobsters of all sizes. They can squeeze into crevices and use their beak to crack my carapace. Seals have been documented eating lobsters, and large crabs occasionally kill and eat small lobsters. Human beings are by far my most significant predator in terms of numbers removed from the population. I host a variety of parasites and symbionts. My shell and gills provide habitat for barnacles, algae, bryozoans, and other encrusting organisms. I tolerate these to a point, but heavy fouling can impair my movement, respiration, and camouflage. I groom regularly, using my small rear legs to scrape my carapace and pleopods, removing some of the fouling organisms. Some of the animals on my shell are commensals, neither harming nor helping me, while others may provide a small benefit, such as additional camouflage. I am also host to internal parasites, including trematodes, nematodes, and protozoans. Most of these cause little obvious harm, but heavy parasite loads can reduce my growth and reproductive success. A particularly visible condition is shell disease, a syndrome that causes lesions and pitting on my carapace, often starting as small dark spots that expand into eroded areas. Shell disease is caused by bacteria, possibly in combination with environmental stress, high temperature, and other factors. It is more common in warmer waters and in polluted areas. Severe cases can lead to infection, impaired molting, and death. The prevalence of shell disease has increased in some parts of my range in recent decades, and researchers are studying its causes and consequences. I communicate with my own kind through multiple channels. Chemical communication is paramount. I constantly release and detect chemical signals in the water. Urine, released from nephropores near the base of my antennae, carries information about my identity, size, sex, reproductive status, and recent fights. I can recognize individuals by their chemical signature and remember past encounters. Dominance relationships are maintained partly through these chemical signals. A subordinate lobster can detect the urine of a dominant individual from meters away and will often avoid the area. During courtship, females release pheromones that signal receptivity, altering the aggressive behavior of males and facilitating pair bonding. I also communicate through visual displays and body postures. Raising my body and claws, called a maral spread, signals aggression and dominance. Lowering my body and folding my claws signals submission. Rapid tail flipping signals alarm and can alert nearby lobsters to danger. I do not produce sound in the conventional sense, but I create water disturbances with my movements, and other lobsters can detect these with their mechanoreceptors. There is some evidence that lobsters produce low-frequency sounds by rubbing parts of their anatomy together, a process called stridulation, though this is not well studied and appears to be less common than in spiny lobsters, which are known for their rasping sounds. Touch is another important mode of interaction. During courtship, male and female touch antennae, claws, and bodies in what appears to be a ritualized assessment. During fights, the physical contact itself conveys information about the opponent's strength and willingness to escalate. Juveniles and sometimes adults engage in brief tactile interactions, touching antennae or claws, which may serve to establish recognition or reinforce social memory. My daily routine varies with season. In summer, when water is warm and food is abundant, I am active nearly every night. I leave my shelter at dusk, forage for several hours, and return before dawn. I may move several hundred meters in a night, following scent trails and exploring new areas. During the day, I rest in my shelter, occasionally grooming my antennae, legs, and shell, using specialized CD and small pincers to remove debris and parasites. Grooming is important for maintaining my sensory structures and for preventing fouling organisms from overgrowing my gills and joints. In autumn, as water temperature begins to drop, my activity gradually declines. I may forage less frequently, and I spend more time in my shelter. In the northern parts of my range, winter brings near-freezing water temperatures. I become sluggish and inactive, entering a state of reduced metabolic activity, sometimes called torpor, though it is not a true hibernation. I do not feed or feed only rarely, and I remain in my shelter for weeks at a time. My heart rate and respiration slow, and I survive on stored energy reserves. In the southern parts of my range, where winters are milder, this winter dormancy is less pronounced, and I may continue to forage sporadically throughout the winter. Spring brings warming water and increasing day length. I resume activity, feeding heavily to rebuild energy reserves depleted over winter. Spring and early summer are often times of increased movement and interaction. Mature males begin to seek out females, and females carrying eggs may release their larvae. The timing of reproduction and larval release is closely tied to water temperature and the seasonal plankton bloom. Larvae released in late spring or early summer encounter abundant planktonic food, increasing their chances of survival. My movements over larger spatial scales vary. Some lobsters are highly sedentary, remaining within a small home range of a few hundred square meters for months or years. Others are more mobile, moving several kilometers over the course of a season. These movements are often related to temperature and food availability. In some regions, particularly in the Gulf of Maine, there is evidence of seasonal inshore-offshore migrations. In autumn, as coastal waters cool, some lobsters move offshore to deeper, warmer water. In spring, they return to shallower, warmer inshore waters. These migrations are not undertaken by all individuals. Many remain in the same area year-round. The factors that determine whether an individual migrates or remains resident are not fully understood, but size, sex, and local environmental conditions likely play roles. Navigation during these migrations may rely on a combination of chemical cues, current direction, water temperature gradients, and possibly geomagnetic or celestial cues, though the mechanisms are still being investigated. My relationship with human beings is ancient and complex. Coastal peoples have harvested lobsters for thousands of years. Archaeological evidence from shell middens, ancient garbage heaps, along the North American coast shows that indigenous peoples consumed lobsters long before European contact. Early European settlers in North America found lobsters so abundant that they were sometimes used as bait, fertilizer, and food for prisoners and servants. Lobsters were considered a low-status food, a perception that persisted until the 19th century. The development of canning and improved transportation changed this. By the mid-1800s, lobster was being shipped inland and overseas, and demand increased. Overfishing soon followed. By the early 20th century, lobster populations in many areas had declined dramatically due to unregulated harvest, habitat destruction, and pollution. Management efforts began in the late 1800s and early 1900s. Regulations were introduced, minimum size limits to protect juveniles, prohibition on taking egg-bearing females, limits on harvest seasons, and gear restrictions. These measures have been refined and expanded over the decades. In most jurisdictions where I am harvested, there are now strict rules. Fishers must use traps, also called pots, baited with fish and designed with escape vents to allow undersized lobsters to exit. Each trap is marked with a buoy, and fishers check their traps every few days, removing legal-sized lobsters and returning undersized individuals and egg-bearing females to the water. Minimum size limits, measured as carapace length, typically range from about 80 to 90 millimeters, depending on jurisdiction. Maximum size limits have also been introduced in some areas to protect the large, highly fecund breeding females. V-notching programs mark egg-bearing females by cutting a small notch in the tail flipper, and these notched individuals are protected from harvest even after they have released their eggs, on the assumption that they will breed again. The fishery for my species is one of the most valuable in North America. In recent decades, landings have reached record highs, particularly in the Gulf of Maine and Atlantic Canada. This increase has been attributed to several factors: effective management, warming water temperatures that favor my growth and reproduction in northern areas, and the decline of predatory fish such as cod, which used to eat large numbers of juvenile lobsters. However, the long-term sustainability of these high harvest levels is uncertain. Climate change is altering ocean temperatures, chemistry, and currents in ways that are difficult to predict. Warming water in the northern parts of my range may continue to benefit lobster populations in the short term, but further warming could exceed my thermal tolerance and favor diseases and competitors. In the southern parts of my range, particularly in southern New England, lobster populations have declined sharply in recent decades, likely due to warming water and increased stress, disease, and reduced recruitment of juveniles. Ocean acidification, driven by increasing atmospheric carbon dioxide, is another emerging threat. Acidification reduces the availability of carbonate ions, which I need to build my shell. Laboratory studies suggest that lobster larvae and juveniles may be particularly vulnerable, experiencing slower growth, thinner shells, and higher mortality in acidified water. The full impact of ocean acidification in the wild is still being studied. Habitat loss and degradation also affect me. Coastal development, dredging, and bottom trawling destroy the rocky and structured habitats I depend on. Pollution from agricultural runoff, sewage, and industrial discharges can degrade water quality, reduce oxygen levels, and increase the prevalence of disease. Plastics and other debris can entangle me or be ingested, though the extent of these impacts is not well quantified. There are conservation measures in place, and in many regions, lobster populations are considered healthy and well managed. The fishery is closely monitored, and regulations are adjusted based on scientific assessments of population status. However, management is complex, involving multiple jurisdictions, political and economic pressures, and scientific uncertainty. The future health of lobster populations will depend on continued effective management, mitigation of climate change and pollution, and protection of critical habitats. There are misconceptions about me that persist in popular culture. One common myth is that lobsters mate for life. This is false. As I have described, mating involves temporary pair bonds lasting only a few weeks, and both males and females may mate with multiple partners over their lifetimes. Another misconception is that lobsters scream when placed in boiling water. I do not have vocal cords or lungs, and I cannot produce a scream. The sound sometimes heard when a lobster is boiled is caused by air and steam escaping from the shell, not vocalization. There is ongoing debate about whether I experience pain in the way vertebrates do. I have a decentralized nervous system, and the question of whether I have subjective experience, consciousness, or the capacity to suffer is a matter of scientific and ethical discussion. I respond to noxious stimuli with avoidance behaviors, which suggests I detect and react to harmful conditions, but whether this constitutes pain as humans understand it is not resolved. Some jurisdictions have enacted welfare regulations requiring that lobsters be stunned or killed quickly before cooking, reflecting evolving ethical considerations. Another misconception is that I am biologically immortal. This idea stems from the observation that I can live a long time, continue to reproduce throughout life, and regenerate lost limbs. Some popular articles have claimed that lobsters do not age and could live indefinitely if not killed by predators or disease. This is an oversimplification. While I do not show some typical signs of aging, such as a decline in reproductive capacity or an exponential increase in mortality rate with age, I am not immortal. I do age. My telomeres, the protective caps on chromosomes, do not shorten as dramatically as in many animals because I produce an enzyme called telomerase throughout life. But this does not mean I am immune to the accumulated damage of living. Very old lobsters may become less able to successfully complete molts, more prone to shell disease, and more vulnerable to predation. Eventually, I will die, whether from disease, injury, predation, or the cumulative effects of age. The claim of immortality is a myth. Let me describe a year in my life as an adult male, about 15 years old, living off the coast of Maine. It is early April. The water temperature is still cold, around 4 degrees Celsius, and I have been largely inactive through the winter, sheltering under a large boulder in 8 meters of water. My shelter is a space I excavated last autumn, scraping away sand and small stones to create a burrow that extends back under the boulder. I share this area with several other lobsters, each in their own shelter spaced tens of meters apart. We are familiar with one another through past encounters, and a loose dominance hierarchy exists. I am a large male, nearly 40 centimeters in total length, and I dominate most of the others nearby. As the days lengthen and the sun angle increases, the water begins to warm. By late April, the temperature has risen to 7 degrees, and I become more active. I begin to forage again, emerging from my shelter in the evening and walking slowly across the rocky bottom. Food is scarce at first. The winter has depleted the local populations of prey, but I find the carcass of a sea urchin, broken and partially eaten by something else, and I consume it. Over the following nights, I range farther, sometimes 200 meters from my shelter, following scent plumes. I find a dead fish, a small sculpin, and I tear it apart with my cutter claw and feed. By mid-May, the water has warmed to 10 degrees and the activity on the bottom has increased. Crabs are emerging from winter torpor, juvenile fish are moving into shallow water, and the seaweed beds are beginning to grow. I feed nightly, catching green crabs, small rock crabs, and the occasional blue mussel pried from a rock crevice. I am also preparing to molt. I have not molted in nearly 2 years, and my shell is encrusted with barnacles, algae, and small bryozoans. My body has grown within the confines of the old shell, and the pressure is building. I can feel the loosening of the carapace at the edges, a sign that the hormonal processes are underway. In early June, I stop feeding entirely. I retreat to my shelter and remain there. Over the course of a week, I become increasingly inactive, my movements reduced to occasional shifts in position. Then one night, the molt begins. My carapace splits along the back seam, and I begin the slow, laborious process of pulling myself out. It takes nearly 2 hours. My claws are the hardest part. They are massive, and the old claw shell is tight. I must compress the claw muscle and pull each claw through the narrow opening of the old shell. Finally, I am free, lying beside the ghostly husk of my old exoskeleton. I am pale, soft, wrinkled, and utterly vulnerable. I take up water, swelling my body and stretching the new shell to a size perhaps 10% larger than the old. My new carapace is thin and flexible like parchment. I remain motionless in my shelter, claws drawn in, antennae drooping. I am helpless. If a predator finds me now, I will die. For 9 days, I do not leave my shelter. Gradually, my shell hardens. The cuticle secretes chitin and begins to deposit calcium carbonate. My color darkens from pale green to the darker mottled pattern of an adult. My claws regain their rigidity. By the 10th day, my shell is hard enough to function, though it will continue to harden for several more weeks. I venture out cautiously, testing my new body. I am larger now, my claws slightly more formidable, and my status in the local hierarchy may have shifted. I encounter a smaller male, one who had been a near equal before my molt. He approaches and we engage in a brief contest, jetting urine at each other, raising claws. He retreats. I have reasserted dominance. June and July are the height of the feeding season. The water has warmed to 15 degrees, my metabolism is high and food is abundant. I forage aggressively, catching crabs, fish, sea urchins, and scavenging any carrion I find. I also encounter females. Some are carrying eggs and I ignore them. They are not receptive and they actively avoid males. Others have recently released their larvae and are beginning to emit pheromones signaling their upcoming molt and receptivity. I detect one such female as she approaches my territory. Her chemical signal is clear and compelling. Over several nights, she visits my shelter, and we engage in a ritualized courtship, touching antennae, sparring gently. She is cautious, testing my response, and I, driven by her pheromones, tolerate her presence in a way I would tolerate no other lobster. One evening, she enters my shelter and does not leave. She positions herself near the back, and I remain near the entrance, facing outward. 2 days later, she molts. I do not help her, but I guard the entrance, fending off a smaller male who attempts to intrude. Once she is soft, I mate with her, depositing spermatophores. She remains with me for 12 days, and during this time I continue to forage, but I return frequently to check on her. By the time she leaves, her shell is hardening, and she moves off to establish her own territory. I will not see her again, though she carries my sperm and will eventually produce larvae sired by me. August brings the warmest water of the year, sometimes exceeding 18 degrees. I am highly active, but I also begin to feel stress. The water holds less oxygen at this temperature, and my gills work harder. I stay in deeper, cooler water during the day and move into shallower areas at night when the water cools slightly. I avoid the warmest patches and the shallowest tide pools. Shell disease is more common now. I see several lobsters with lesions on their carapaces, dark spots that have begun to erode the shell. I am fortunate. I have no lesions yet, perhaps because my shell is newly hardened and clean. By September, the water begins to cool again and the stress of high temperature eases. I feed heavily, building energy reserves for the coming winter. The abundance of prey begins to decline as crabs and other invertebrates become less active and some fish migrate offshore. I focus on mussels, clams, and sea urchins, which are still available. October brings shorter days and dropping temperatures. By late October, the water is down to 10 degrees, and I begin to reduce my activity. I have located a good winter shelter, a deep crevice under a large rock ledge in slightly deeper water about 12 meters, where the temperature will be more stable. I move to this shelter in early November, and I prepare for winter dormancy. December through March are the quiet months. The water temperature hovers between 2 and 4 degrees. I remain in my shelter almost constantly, occasionally shifting position or extending my antennae to monitor the surrounding water, but I do not forage. My metabolic rate is at its lowest, and I survive on the glycogen and lipid reserves I accumulated in summer and autumn. Snow falls on the surface above and storms churn the water, but at 12 meters depth the disturbance is muted. I am in a state of waiting, my life reduced to basic maintenance, breathing slowly, heart beating faintly, a living statue in the cold darkness. As April returns, the cycle begins again. The water warms, I stir, and I emerge once more into the world of movement, hunger, competition, and reproduction. This pattern has repeated for 15 years of my life, and if fortune holds, it may repeat for many more. Now let me shift perspective and describe the life of a female, focusing on the remarkable journey of reproduction and maternal care. I am a female lobster, approximately 12 years old, weighing just over 1 kilogram. I live in a rocky area off the coast of Nova Scotia, in water ranging from 5 to 15 meters deep depending on season. 2 years ago, I mated with a large male after molting in his shelter. I have been carrying his sperm in my seminal receptacle ever since, waiting for the right conditions to extrude and fertilize my eggs. It is now early summer, June, and the water has warmed to 12 degrees. I have been feeding heavily for weeks, accumulating energy. My ovaries, located in my thorax, have been growing, filled with developing oocytes. The hormonal signals have aligned. Day length, temperature, my nutritional state, all indicating that now is the time. One evening, I position myself in my shelter and I begin to extrude eggs. The eggs emerge from the gonopores, small openings on the third segment of my thorax, as I curve my tail forward beneath my body. As each egg emerges, it is fertilized by sperm released from my seminal receptacle and is immediately glued to one of my pleopods by a sticky secretion. This process takes several hours, and by the time I am finished, I am carrying approximately 40,000 eggs, each a dark greenish bead, covering the underside of my tail in a dense, lumpy mass. For the next 11 months, I am a buried female. My behavior changes. I am more cautious, less likely to engage in aggressive encounters. I forage, but I stay closer to my shelter and avoid areas where I might encounter large predators or aggressive males. Every day I groom my eggs, using my rear walking legs to pick through the mass, removing any eggs that have died or become fouled. I fan them with my pleopods, creating a gentle current of water that brings oxygen and removes carbon dioxide and ammonia. This grooming is time-consuming and essential. If I neglect it, fungal infections will spread and I will lose the brood. Winter comes and I reduce my activity, but I do not stop caring for the eggs. Even in the coldest months when I am otherwise dormant, I continue to fan and groom them, though less frequently. The eggs develop slowly in the cold water. By early spring, they have darkened, and I can see the tiny eye spots of the embryos through the translucent egg membranes. In late May of the following year, Nearly 12 months after I extruded the eggs, they are ready to hatch. I move to an area with good current flow, a rocky outcrop where the water is well oxygenated and the current will carry the larvae away from the concentration of bottom predators. Over the course of a week, the eggs begin to hatch. The larvae wriggle free from the egg membrane and immediately swim upward, away from me. I do not protect them or provision them. My role ends at hatching. I release them in pulses, thousands at a time, usually at night. By the end of the week, all the eggs have hatched and my pleopods are bare. I feel lighter, more agile. I can now perform the rapid tail flip escape again without the burden of the egg mass. I have not molted in 2 years since before I mated. My shell is fouled and tight. Within a few weeks of releasing my larvae, I begin to prepare for a molt. I stop feeding, retreat to my shelter, and go through the process I have described. I emerge from the old shell, soft and vulnerable, and I remain hidden for over a week while the new shell hardens. I have not mated again. I will not be receptive for another year or more, depending on my condition and the environmental conditions. Meanwhile, the larvae I released are in the plankton. Each one is on its own, drifting with the currents, feeding on microscopic prey, growing and molting through the 3 planktonic stages. Most of them will die. They are eaten by fish, by jellyfish, by predatory plankton. Some will starve if they do not find sufficient food. Some will be carried by currents into unsuitable water, too warm, too cold, too fresh, or simply too far from suitable settlement habitat. But a few, perhaps a few dozen out of the 40,000 I released, will survive to the post-larval stage. They will find good cobble habitat, settle, and begin their benthic life. If they are very fortunate, and if conditions are favorable, a handful of those may survive to adulthood and reproduce, continuing the cycle. This is the rhythm of my life as a female, feeding and growing, then molting and mating, then carrying and tending eggs for nearly a year, then releasing larvae and molting again. The entire reproductive cycle takes at least 2 years, often longer. I may repeat this cycle many times over my life, perhaps a dozen times or more if I live to be old. Each time I invest enormous energy, and each time the odds are against any individual offspring surviving. But by producing tens of thousands of eggs over my lifetime, I ensure that a few will succeed, and that is enough. Let me now explore the early life in greater detail, from the perspective of a larva. I have just hatched, wriggling free of the egg membrane, and I swim upward with rapid beats of my pleopods. The sunlight is above, filtering down through the water in shimmering columns of green and blue. I am tiny, transparent, vulnerable, but I am driven by instinct to reach the surface layers where food is concentrated. Within hours, I am in the upper 10 meters of the water column, among a dense soup of plankton, copepods, larval barnacles, diatoms, dinoflagellates, and countless other drifting organisms. I am hungry. My yolk reserves, carried over from the egg, are exhausted. I must eat. I detect a copepod, a small crustacean, by the water disturbances it creates. I lunge, my tiny claws grasping, and I capture it. I bring it to my mouth and consume it. Over the following days, I feed continuously, capturing and eating copepods and other small prey. I grow rapidly, and within a week, I undergo my first larval molt. My body lengthens, my spines become more pronounced, and my appendages more developed. I am at the mercy of the currents. I have some ability to swim vertically, moving up and down in the water column, and there is evidence that lobster larvae use this ability to position themselves in currents that will carry them towards suitable settlement areas, though the mechanisms are not fully understood. I drift for weeks. The water temperature, the abundance of food, and the presence of predators all affect my survival. I am attacked repeatedly. A small jellyfish brushes me with a tentacle, and I barely escape. A larval fish, no bigger than I am, tries to eat me, and I thrash and swim away. A comb jelly engulfs me, but I am too large and spiny, and it expels me. After 3 weeks, I molt again, entering the third larval stage. I am larger, more robust, but still planktonic. My eyes are more developed, my claws more functional. I continue to feed and grow. Another 2 weeks pass, and I molt a third time, becoming a post-larva. I now look like a tiny lobster with a segmented abdomen, a carapace, and small but functional claws. I'm about 12 millimeters long. My behavior changes. I begin to dive, swimming down to explore the bottom, then swimming back up. I repeat this over and over, sometimes dozens of times. I am searching for the right place to settle. On one of these dives, I reach the bottom in an area of cobble and small boulders, with patches of algae and a scattering of broken shells. This looks right. I land on the bottom and scuttle into a tiny crevice between 2 stones. It is a tight fit, and I am hidden from view. I remain there for several hours, testing the site. The water flow is good, the temperature is tolerable, and I sense no immediate danger. I decide to stay. I molt one more time, becoming a benthic juvenile, and I begin my life on the bottom. The next several years are a time of hiding, growing, and learning. I venture out only briefly to feed, and I return quickly to my shelter. I eat detritus, tiny worms, bits of algae, and the occasional microscopic crustacean. I molt every few weeks, each time growing larger and needing a new shelter. I am almost always hungry, and I am surrounded by predators. Small fish, crabs, and other lobsters all see me as prey. I learn to recognize the chemical signatures of danger, the scent of a predator, the urine of a dominant lobster, and I learn to avoid these signals. I learn the locations of good shelters, the places where food is often found, and the times of day when predators are less active. By the time I am 3 years old, I am 5 centimeters long and beginning to look like a small adult. I have survived the most dangerous phase of my life. My shell is harder, my claws more capable, and I can defend myself against some threats. I am no longer easy prey for most fish, though I am still vulnerable to octopuses, large crabs, and bigger lobsters. I continue to grow, to molt, and to feed. I establish a small territory, a burrow under a rock that I defend against other juveniles. I begin to participate in the social interactions of adult lobsters, though I am still small and subordinate to most others. At 6 years old, I reach sexual maturity. I am a male, about 20 centimeters long, weighing perhaps 200 grams. I have survived against long odds, and I am now capable of reproducing. I will compete with other males for access to females, and if I am successful, I will pass my genes to the next generation. The cycle continues. The ecological importance of lobsters extends beyond our direct interactions with prey and predators. We are ecosystem engineers in a small way. Our burrowing and excavation of shelters disturb the sediment, creating microhabitats for other organisms. Small fish, crabs, and invertebrates sometimes share or take over old lobster burrows. Our feeding reduces the abundance of sea urchins and other grazers, which can have cascading effects on seaweed and kelp forests. In areas where lobster populations have been depleted, urchin populations have sometimes exploded, leading to overgrazing of kelp and the creation of urchin barrens, rocky areas almost devoid of seaweed. Where lobster populations are healthy, urchins are kept in check, and kelp forests thrive, supporting diverse communities of fish and invertebrates. We are also an important link in nutrient cycles. We consume a wide range of organic material, from live prey to detritus and carrion, and we convert this into lobster biomass and waste products. Our feces and molted shells contribute organic matter and calcium to the sediment, where it is further processed by bacteria and detritivores. When we die, our bodies are scavenged by other lobsters, crabs, fish, and invertebrates, returning nutrients to the food web. Our interactions with humans are not limited to fishing. Lobsters have become important in scientific research. We are used as model organisms to study topics as diverse as neurobiology, aggression, hormonal control of molting, sensory biology, and the effects of environmental stressors. Research on lobster neurons has contributed to fundamental understanding of how nervous systems work, and studies of lobster social behavior have provided insights into the evolution of dominance hierarchies and communication. We are also subjects of conservation and management research, as scientists work to understand population dynamics, recruitment variability, and the impacts of climate change and fishing pressure. Culturally, lobsters have a complex place in human societies. In North America, lobster is now considered a delicacy, associated with luxury and fine dining, though this is a relatively recent shift. Lobster festivals, lobster shacks, and the iconic image of a whole steamed lobster with melted butter are part of the regional identity of coastal New England and Atlantic Canada. In Europe, Homarus gammarus holds a similar cultural place in countries like France, the United Kingdom, and Norway. Lobsters appear in literature, art, and popular media, sometimes as symbols of the sea, sometimes as humorous or curious creatures due to our unusual appearance and behavior. There are ethical and philosophical questions raised by our harvest and treatment. As I mentioned earlier, the question of whether lobsters feel pain is unresolved and is the subject of ongoing scientific investigation and ethical debate. Some animal welfare advocates argue that lobsters should be afforded protections similar to those given to vertebrate animals, including requirements for humane slaughter. Others argue that our nervous systems are sufficiently different from those of vertebrates that the concept of pain may not apply in the same way. Research into crustacean sentience is ongoing, and societal attitudes and regulations may continue to evolve as scientific understanding improves. Climate change is perhaps the most significant long-term challenge facing lobster populations. Warming ocean temperatures are already shifting the distribution of American lobsters northward and into deeper water. In southern New England, where water temperatures have increased by more than 1 degree Celsius over recent decades, lobster populations have declined dramatically. The warming has led to increased stress, higher prevalence of shell disease, reduced reproduction, and poor survival of juveniles. In contrast, in the Gulf of Maine and in Canadian waters, where temperatures have also warmed but remain within the tolerable range for lobsters, populations have increased, and the fishery has been exceptionally productive. However, continued warming is predicted to eventually exceed the thermal tolerance of lobsters even in these northern areas, and future population trends are uncertain. Ocean acidification, the result of increasing carbon dioxide dissolving in seawater and lowering pH, poses a chemical challenge. Lobster larvae, with their thin, delicate shells, are particularly vulnerable. Studies have shown that larvae reared in acidified water grow more slowly, have weaker shells, and suffer higher mortality. Adult lobsters appear somewhat more resilient, but chronic exposure to low pH may impair shell formation, reproduction, and overall fitness. The combined effects of warming and acidification are likely to be complex and difficult to predict, and they represent a major area of ongoing research. Changes in ocean currents and stratification, also linked to climate change, may affect the dispersal and settlement of lobster larvae. If currents shift, larvae may be carried away from suitable settlement habitats, reducing recruitment and leading to population declines. Changes in the timing and intensity of plankton blooms, driven by warming and changing nutrient dynamics, may affect the availability of food for larvae, further influencing survival. Human-caused pollution continues to threaten lobster habitats. Nutrient runoff from agriculture and sewage can lead to eutrophication, algal blooms, and oxygen-depleted dead zones. Heavy metals, pesticides, and industrial chemicals can accumulate in sediments and in lobster tissues, potentially affecting health and reproduction. Microplastics are increasingly pervasive in the ocean, and there is evidence that lobsters and other crustaceans ingest these particles, though the long-term consequences are not yet well understood. Habitat protection is critical for the future of lobster populations. Coastal development, dredging, and destructive fishing practices such as bottom trawling can destroy the complex rocky habitats that lobsters depend on. Marine protected areas, where fishing and other extractive activities are limited or prohibited, can serve as refuges for lobsters and other species, allowing populations to recover and supporting ecosystem health. Some regions have established lobster conservation zones, areas where lobster fishing is restricted to protect breeding populations and habitats. The effectiveness of these measures depends on enforcement, compliance, and the scale of protection relative to the mobility and life history of lobsters. Genetic diversity is another important consideration for long-term population resilience. Lobster populations are connected by larval dispersal, and there is gene flow among regions, but there is also evidence of genetic structure, with populations in different areas showing subtle genetic differences. Overfishing in one area can reduce genetic diversity locally and potentially reduce the adaptive capacity of the population to respond to environmental change. Management strategies that maintain population size and connectivity are important for preserving genetic health. Research into lobster aquaculture has been ongoing for decades. Culturing lobsters from eggs to market size is technically feasible, but it remains economically challenging. Lobsters grow slowly, require high-quality water and food, and are cannibalistic, requiring individual rearing or very low densities. These factors make aquaculture expensive compared to wild harvest. However, as wild populations face increasing pressures, and as techniques improve, aquaculture may play a larger role in supplying demand and reducing pressure on wild stocks. Some hatcheries produce juvenile lobsters for stock enhancement, releasing them into the wild to supplement natural recruitment, though the effectiveness of these programs is debated and requires careful monitoring. There is also interest in using lobsters and other shellfish in ecosystem-based management and restoration. In some regions, efforts are underway to restore degraded coastal habitats, including oyster reefs, eelgrass beds, and kelp forests. Healthy lobster populations can be indicators of ecosystem health, and conversely, the recovery of lobster populations may signal broader ecosystem recovery. Lobsters, as predators and prey, are integral to the food webs of coastal ecosystems, and their conservation is linked to the conservation of the entire system. In terms of disease management, understanding and controlling shell disease and other pathogens is a priority. Research is focused on identifying the causative agents of shell disease, understanding the environmental and physiological factors that increase susceptibility, and developing potential treatments or management practices to reduce prevalence. Warming water temperatures appear to increase the risk of shell disease, and areas with high fishing pressure and dense lobster populations may also be more prone to outbreaks. Managing these factors may help reduce disease impacts. Interdisciplinary research is increasingly important. Ecologists, oceanographers, geneticists, fisheries scientists, climate scientists, and social scientists are working together to understand the complex interactions between lobsters, the environment, and human activities. Models that integrate biological, physical, and socioeconomic data are being developed to predict future population trends and to evaluate the potential outcomes of different management strategies. These models are tools to help managers and policymakers make informed decisions, though they are inherently uncertain and must be continually updated as new data become available. Public engagement and education are also crucial. Many people who consume lobster or live in coastal areas have little understanding of lobster biology, ecology, or the challenges facing populations. Outreach efforts, including educational programs, citizen science initiatives, and public communication by scientists and managers, can increase awareness and support for conservation measures. Sustainable seafood programs, which certify fisheries that meet certain environmental and management standards, can help consumers make informed choices and reward fisheries that are well managed. From my perspective as a lobster, the future is uncertain. I do not contemplate the future in the human sense, but my survival and the survival of my offspring depend on the conditions of the ocean and the actions of human beings. The ocean has been my home for 100 million years, through ice ages and warm periods, through asteroid impacts and mass extinctions. My lineage has persisted, adapted, and thrived. But the current rate of environmental change is unprecedented in the history of my kind. Whether lobsters will continue to thrive in the oceans of the coming decades and centuries depends on factors beyond my control. The trajectory of climate change, the effectiveness of management and conservation efforts, and the broader health of ocean ecosystems. I will continue to do what I have always done. Hide in crevices, hunt in the darkness, fight for mates and territory, molt and grow, reproduce and release larvae into the plankton. I am a creature of instinct and adaptation, shaped by millions of years of evolution to live in the cold, rocky, dynamic world of the ocean floor. I am part of a vast web of life, connected to seaweeds and microbes, to fish and seals, to the currents and the tides, to the sun and the seasons. My story is the story of survival, of persistence, of the intricate and often hidden lives of the ocean's inhabitants. Let me return once more to the sensory world I inhabit, because it is so different from the human experience and so central to how I navigate my life. I live in a world of gradients, temperature gradients, salinity gradients, light gradients, and especially chemical gradients. My primary sense is chemical. The water around me is a flowing medium carrying dissolved molecules from every source. Decaying organisms, living prey, predators, conspecifics, algae, bacteria, sediments. I am constantly sampling this chemical landscape with my antennules, mouthparts, and leg tips. I can detect amino acids at concentrations of parts per billion, and I can distinguish among different compounds, identifying the signature of a crushed crab versus a dead fish versus the urine of a male lobster versus a female. This is not smell or taste in the human sense. It is a direct chemical sense, immediate and spatial. I can determine the direction of a chemical source by comparing the concentration detected by my left and right antennules, and I can follow a plume upstream by walking into the current and adjusting my path as the concentration increases or decreases. My mechanoreception is also acute. My body is covered with seedy, hair-like structures that are sensitive to water movement. I can detect the approach of a predator by the pressure wave it creates, the vibrations of a struggling prey item, the current patterns around rocks and obstacles. My antennae are long and flexible, and I sweep them constantly, feeling the contours of the bottom, the texture of surfaces, and the presence of objects. When I walk, my antennae are in constant motion, providing a tactile map of the immediate environment. My vision, as I have mentioned, is adapted to the dim and shifting light of the seafloor. I see movement well, and I can detect changes in light intensity, which helps me distinguish day from night and recognize the silhouette of a predator passing overhead. My compound eyes give me a wide field of view, nearly 360 degrees when I include the movement of my eye stalks, but the resolution is low. I do not see fine details or recognize faces. However, I can detect the polarization of light, which is a feature of sunlight scattered by water and the sky. This may help me orient myself, determine the direction of the sun even on overcast days, and navigate during migrations. I do not hear in the way that humans or fish do. I do not have ears or a swim bladder to detect pressure waves. However, I am sensitive to low-frequency vibrations transmitted through the water and the substrate. My legs and body can detect these vibrations, and I may use them to sense approaching predators or the activity of other lobsters. Some research suggests that crustaceans, including lobsters, can detect and respond to underwater sound, though the mechanisms and the ecological significance are not well understood. I have a sense of balance and orientation provided by statocysts, small organs located at the base of my antennules. Each statocyst is a fluid-filled sac containing a statolith, a small calcareous structure, and sensory hairs. As I move and tilt, the statolith shifts, bending the hairs and providing information about my orientation relative to gravity. This is essential for coordinating my movements, for righting myself if I am flipped over, and for executing the tail flip escape response in the correct direction. My internal state is regulated by a complex interplay of hormones and neurotransmitters. My molting cycle is controlled by hormones produced in the X-organ and sinus gland, located in my eye stalks, and in the Y-organ, located in my thorax. The balance between molt-inhibiting hormone and ecdysteroids, the molting hormones, determines when I molt. My reproductive state is also hormonally controlled, with influences from environmental cues such as temperature and photoperiod. My aggression and social behavior are modulated by neurotransmitters including serotonin and octopamine. Research has shown that injecting serotonin into a lobster can increase dominant, aggressive behavior, while octopamine is associated with submissive behavior. This has led to fascinating studies on the neurochemical basis of social hierarchies and behavior, and lobsters have become a model for understanding the evolution of these systems across the animal kingdom. I am capable of learning and memory, though the extent and mechanisms are still being studied. I can learn to associate certain stimuli with food or danger, and I can remember the locations of shelters and the identities of individual lobsters I have encountered. Some experiments have shown that lobsters can learn to navigate mazes and can retain this learned information for days or weeks. My memory of past fights appears to influence my future behavior. If I have lost a fight to a particular individual, I am more likely to avoid that individual in future encounters, even if the encounter occurs days later and in a different location. This suggests that I have some form of social memory, recognizing individuals by their chemical signatures and recalling the outcome of previous interactions. Pain and stress responses in lobsters are areas of active research and debate. I respond to noxious stimuli, such as extreme temperature, acidic or basic solutions, and physical injury, with avoidance behaviors and physiological changes. My heart rate increases, stress hormones are released, and I exhibit behaviors that suggest I am trying to escape the source of the stimulus. Some researchers interpret these responses as evidence of a pain-like state, while others caution that behavioral and physiological responses to harmful stimuli do not necessarily indicate subjective experience of suffering, especially in an animal with a decentralized nervous system, so different from that of vertebrates. The question hinges partly on definitions. What is pain? What is suffering, and what neural substrates are necessary for these experiences? These are difficult questions, and they sit at the intersection of biology, philosophy, and ethics. What is clear is that I respond to my environment in complex ways, that I have mechanisms to detect and avoid harm, and that my welfare, however defined, can be affected by how I am treated. The management of lobster fisheries involves a complex set of regulations, monitoring, and enforcement efforts. In jurisdictions such as the state of Maine, the Canadian Atlantic provinces, and European countries, lobster fisheries are subject to rules that have been developed and refined over more than a century. These rules include minimum and sometimes maximum size limits, measured as carapace length, protection of egg-bearing females, often enforced through V-notching programs, seasonal closures in some areas, restrictions on gear type and configuration, such as requirements for escape vents in traps and biodegradable panels that allow lobsters to escape from lost or abandoned traps, limits on the number of traps each fisher can deploy, and licensing systems that control who can participate in the fishery. Enforcement is carried out by Marine Patrol officers, Coast Guard and fisheries officers, who conduct inspections at sea and at docks, check licenses and gear, measure lobsters to ensure compliance with size limits, and check for V-notched females. Violations can result in fines, loss of licenses, and criminal charges in severe cases. Compliance is generally high in well-managed fisheries, partly because the fishers themselves have a long-term interest in sustaining the resource, and partly because community norms and peer pressure discourage poaching and illegal practices. Data collection is a critical component of management. Fisheries scientists conduct surveys to estimate lobster abundance, size distribution, and reproductive status. These surveys use a variety of methods, including trap surveys, where standardized traps are deployed in a grid pattern and the catch is recorded, dive surveys, where divers count and measure lobsters in specific areas, and trawl surveys, where nets are dragged along the bottom to sample the lobster population along with other species. Data on commercial catch are also collected, including total landings, size composition, and effort, measured as the number of traps deployed and the number of days fished. These data are analyzed to assess population trends, estimate recruitment of juveniles into the fishery, and determine whether current fishing pressure is sustainable. Stock assessment models are used to synthesize all this information and to provide advice to managers. These models are complex, incorporating data on population size, growth rates, mortality rates, recruitment, and fishing effort. They attempt to predict future population trends under different management scenarios, such as changes in size limits, trap limits, or seasonal closures. The models have inherent uncertainties due to natural variability in recruitment, environmental factors, and limitations in the data. Managers use the model outputs along with socioeconomic considerations, stakeholder input, and precautionary principles to set regulations. There is increasing recognition that lobster management must be adaptive, meaning that regulations are adjusted in response to new data and changing conditions. Climate change, shifts in predator and prey populations, and changes in fishing technology and markets all require ongoing attention and flexibility. Some jurisdictions have implemented adaptive management frameworks that include regular review of data, stakeholder involvement in decision-making, and mechanisms to respond quickly to signs of population decline or other problems. International cooperation is also important, especially for species like the American lobster, whose range spans multiple national and subnational jurisdictions. Lobsters do not recognize political boundaries, and larvae can drift across borders, meaning that populations in one area can be affected by management decisions in another. Canada and the United States work together through various bilateral mechanisms to share data, coordinate research, and discuss management approaches. Within the United States, the Atlantic States Marine Fisheries Commission coordinates management among the states along the Atlantic coast. These collaborative efforts are essential for effective management of a shared resource. There are ongoing debates and challenges in lobster management. One issue is the allocation of fishing rights. In many areas, access to the lobster fishery is limited, and licenses are valuable and sometimes difficult to obtain. Questions about who should have access, how to balance the interests of commercial fishers, recreational fishers, and indigenous communities, and how to address the entry of new participants are complex and often contentious. Another issue is the impact of climate change, which is shifting lobster populations and creating winners and losers among fishing communities. Areas that historically had productive lobster fisheries may see declines, while other areas may see increases, leading to social and economic disruption. Another challenge is the bycatch and interactions with other species and gear types. Lobster traps can entangle marine mammals, sea turtles, and seabirds, though the frequency and impact of these interactions are lower than for some other types of fishing gear. Efforts are underway to develop gear modifications and fishing practices that reduce the risk of entanglement. For example, weak links and breakaway panels in trap lines can allow large animals to escape if they become entangled. Seasonal closures in areas where endangered species are present can also reduce interactions. The conservation of North Atlantic right whales, a critically endangered species, has led to significant restrictions on lobster fishing in some areas and times, particularly related to the vertical lines that connect traps to surface buoys. There is active research into ropeless or on-demand fishing systems, where traps do not have a permanent vertical line, but instead are equipped with acoustic release mechanisms that bring the trap to the surface only when the fisher is present. These systems are still being developed and tested, and their feasibility and cost-effectiveness are being evaluated. The relationship between lobster fisheries and broader marine conservation goals is also a topic of discussion. Some conservationists advocate for large marine protected areas where all fishing is prohibited, arguing that these areas are necessary to protect marine biodiversity and ecosystem function. Some fishers and fishery managers are concerned that large closures will displace fishing effort, reduce livelihoods, and may not provide clear benefits for lobster populations if the areas are not well designed. Finding a balance between conservation and sustainable use is an ongoing process that requires dialogue, science, and compromise. From my perspective, none of these human debates and decisions are directly perceptible, but their outcomes determine the conditions of my existence. The number of traps in the water, the size at which I am vulnerable to capture, the protection afforded to egg-bearing females, the health of the ecosystem I live in, all of these are shaped by human choices. My fate and the fate of my kind are intertwined with human society in ways that are unique in the history of life on Earth. Let me describe another aspect of my life, the experience of injury and healing. I live in a physically demanding and dangerous environment, and injuries are common. I may lose a leg or claw in a fight with another lobster, in an encounter with a predator, or caught in a rock crevice during a storm. I may be struck by debris, bitten by a fish, or damaged during a failed molt. When I lose a limb, the wound is initially open, and hemolymph, my blood, leaks out. However, my hemolymph clots quickly, forming a plug that seals the wound within minutes to hours. The clotting process involves cells called hemocytes and a cascade of proteins similar to the clotting systems in other animals. Once the wound is sealed, healing begins. Over the following days and weeks, cells migrate to the site and new tissue begins to form. If the limb was lost at a joint, where there is a natural breakage plane, the healing is usually clean and complete. If the loss was elsewhere, healing may be more complicated and leave a scar. The regeneration of a lost limb is one of my most remarkable abilities. During the next molt after losing a limb, a small limb bud appears at the site. This bud contains undifferentiated cells that will develop into the various tissues of the new limb: muscle, nerve, cuticle. With each subsequent molt, the regenerating limb grows larger, and after 3 or 4 molts, it is usually close to the original size and fully functional. The process is controlled by hormones and growth factors, and it requires significant energy. Regenerating a large claw can reduce my overall growth rate and may delay my sexual maturity or reduce my reproductive output. There is a trade-off between allocating energy to regeneration versus growth or reproduction, and the outcome depends on my nutritional state and environmental conditions. I can also recover from damage to my shell. If my carapace is cracked or punctured, the wound exposes my internal tissues and creates a risk of infection. My hemocytes respond to the injury, and the wound is sealed with a combination of clotted hemolymph and rapid deposition of new cuticle. This patch is often darker or a different texture than the surrounding shell, visible as a scar. At the next molt, the damaged area is shed along with the rest of the old shell, and the new shell is usually intact and unscarred. However, if the damage is severe or if infection sets in, I may die before I can molt. Shell disease, the bacterial erosion of the shell, is a chronic condition that does not heal easily and can worsen over time if environmental conditions do not improve. My immune system is different from that of vertebrates. I do not have antibodies or adaptive immunity. Instead, I rely on innate immunity, physical barriers like my shell, antimicrobial compounds in my hemolymph, and hemocytes that engulf and destroy pathogens. Hemocytes can recognize certain molecular patterns common to bacteria, fungi, and other pathogens, and they respond by engulfing the invader, releasing toxic compounds, and clotting to wall off the infection. This system is effective against many threats, but it is less flexible than adaptive immunity and can be overwhelmed by heavy pathogen loads or novel pathogens to which I have no prior exposure. Parasites and diseases are part of my life. I may host trematode worms that infect my hepatopancreas, nematodes in my muscles, or protozoans in my hemolymph. Most of these parasites cause little obvious harm at low levels, but heavy infections can reduce my growth, reproductive success, and survival. Some parasites alter my behavior. For example, certain trematode infections can make me more sluggish or less responsive, potentially increasing my vulnerability to predation, which may benefit the parasite if its life cycle includes a predator of lobsters. I have no conscious awareness of these parasites, but my body responds to them with immune reactions, sometimes successfully controlling the infection, sometimes not. My relationship with other species extends beyond predation and competition. I interact with a variety of organisms in neutral, commensal, or even mutualistic ways. Small fish sometimes hover near me as I forage, darting in to grab bits of food I disturb or discard. These fish are not harmed by my presence and may benefit from the feeding opportunities I create. Cleaner shrimp and other small crustaceans may pick parasites and debris from my shell and gills, providing a cleaning service that benefits both parties, though the evidence for true mutualism in these interactions is debated. Barnacles and algae that grow on my shell may provide additional camouflage, helping me blend into the rocky substrate, though if the fouling becomes too heavy, it hinders my movement and respiration, and I benefit from molting to shed them. I live within a matrix of interactions, a node in a vast network of feeding, competition, reproduction, and coexistence. My presence affects the abundance and behavior of my prey, and their presence affects me. The predators that hunt me shape my behavior and my evolution. The fastest, most cautious, and most cryptic lobsters are more likely to survive and reproduce, passing on those traits. The other lobsters I compete with for shelter and mates push me to grow larger, to fight more effectively, to read chemical signals more accurately. The parasites and diseases I encounter drive selection for immune function and physiological resilience. The currents and temperatures and chemical conditions of the ocean set the physical stage on which all these interactions play out. From the scale of molecules to the scale of ocean basins, my life is shaped by processes I do not perceive but am embedded within. The genes in my cells, inherited from my parents, encode the instructions for building my body, regulating my metabolism, and responding to my environment. These genes have been shaped by millions of years of natural selection, tested in countless ancestors, refined through survival and reproduction. Mutations arise, some harmful, some neutral, and a few beneficial, and these are sorted by the sieve of survival. I am the product of this process, a living expression of an evolutionary history that stretches back to the first arthropods in the ancient seas. The ocean itself is shaped by forces at planetary scales. The rotation of the Earth, the gravitational pull of the Moon, the energy of the Sun, the chemistry of the atmosphere. Ocean currents transport heat, nutrients, and organisms, connecting distant regions. The tides rise and fall, driven by the Moon's gravity, exposing and submerging the intertidal zones where some of my kind live. The seasons change, driven by the tilt of the Earth's axis, bringing variations in light, temperature, and productivity that ripple through the ecosystem. Climate patterns such as the North Atlantic Oscillation and El Niño influence water temperature, storm frequency, and current patterns over years and decades, affecting the conditions I experience and the survival of my offspring. Human activities are now a planetary force, altering the composition of the atmosphere, the temperature of the ocean, the chemistry of seawater, and the structure of ecosystems. The carbon dioxide emitted by burning fossil fuels has increased the greenhouse effect, warming the planet and the oceans. It has also dissolved in seawater, lowering the pH and reducing the availability of carbonate ions. Nutrients from agriculture, sewage, and industry have altered the productivity and composition of coastal waters. Plastics and other pollutants have spread throughout the ocean, even to the deepest trenches and most remote islands. Fishing has removed enormous quantities of biomass from the sea and has altered the structure of food webs. These changes are happening rapidly, on timescales of decades rather than millennia, and they present challenges that my species has never faced before. Yet life is resilient and adaptable. My ancestors survived mass extinctions, including the event that killed the dinosaurs. They adapted to ice ages and warm periods, to rising and falling sea levels, to the appearance and disappearance of continents. The capacity for adaptation is built into the genetic diversity of populations, the variation in traits among individuals, and the mechanism of natural selection. If environmental conditions change gradually enough, and if genetic diversity is maintained, populations can evolve to match new conditions. But there are limits. If change is too rapid, if populations are too small or isolated, if genetic diversity is too low, adaptation may not occur fast enough, and extinction becomes a risk. The conservation of lobsters and of marine ecosystems more broadly is ultimately about maintaining the conditions that allow life to persist and adapt. It is about protecting habitats, managing harvests sustainably, reducing pollution, mitigating climate change, and preserving the complex web of interactions that makes ecosystems functional and resilient. It is also about recognizing that human well-being is intertwined with the health of the natural world. The ocean provides food, regulates climate, supports livelihoods, and holds intrinsic value as a living system of immense beauty and complexity. From my perspective as a lobster, I do not hope or fear. I do not plan for the future or regret the past. I live in the present, responding to the immediate conditions around me, driven by instincts and reflexes honed by evolution. But my life is part of a larger story, a story of survival and adaptation, of the intricate workings of ecosystems, and of the relationship between humans and the natural world. My story is worth telling not only because I am fascinating in my own right, but because understanding me, how I live, how I grow, how I reproduce, how I fit into the ocean, deepens our understanding of life itself and our place within it. I am a lobster. I walk on the ocean floor, hidden among rocks, hunting and being hunted, molting and growing, fighting and mating, releasing larvae into the plankton, and persisting through seasons and years. I am part of an ancient lineage, a link in a chain of life that stretches back through deep time. I am shaped by my environment and I shape it in turn. I am both ordinary, one individual among billions, and extraordinary, a complex living being with a unique set of experiences and challenges. My life is a testament to the diversity and resilience of life on Earth, and a reminder of the fragility of the systems that sustain us all.